Next speaker is Maria Elena Bottazzi. In an interview with El País in January 2022, she said something that really stayed with me. She said, we need to use vaccine as a tool of decolonization. She was describing the COVID vaccine she was then developing, which was patent-free and especially designed to be easily and cheaply produced in low and middle-income countries. Professor Maria Elena Bottazzi is Associate Dean of the National School of Tropical Medicine at Baylor College of Medicine in Houston and co-director of the Texas Children's Hospital Center for Vaccine Development. Born in Honduras and trained in the United States, she has devoted her career to developing vaccines for neglected tropical diseases that disproportionately affect the world's poorest communities. She also co-led the development of Corbivax, a low-cost, patent-free COVID vaccine. Please welcome Dr. Bottazzi, who will present translational synergies and partnerships to conduct cutting-edge vaccine science innovation.
Thank you. Thank you. Good morning. Good morning. Gracias. It's a pleasure to be here. It was a throwback of memories for me because as much as you're going to hear a little bit of a different talk, little philosophical, maybe social, maybe political, maybe businessy, with a lot of parasites and microbes in between. I actually trained, my postdoc and my PhD actually was all in cellular and molecular biology. So it's like throwback, seeing all the cyclines and all the, it's amazing. So, and thanks to Maria Yanez, because I think it was a little bit her fault. I met her in one of the Spanish extracellular vesicle conferences a couple of years ago. And I think probably she really enjoyed my talk. And I think she pushed to see if I could get invited into this amazing golden Jubilee. So let's see if I can walk you through my presentation. If it works. Oh, it's doing the same thing as yesterday. I think it's the Zoom thing. Let's see. Okay, now it works. Yeah, perfect. All right, so I'm actually bringing you the Texas experience. And why I'm bringing you the Texas experience? Well, first of all, as you can see here in the little map, the red states, they're actually a very unique part of the world. They're actually considered what we call the third coast of the United States. And as much as it's, of course, bordering Mexico, of course, and, you know, it connects us with the rest of Latin America and the Caribbean, it's really also a gateway because, as you know, from there, you can easily go east, go west, go south, right, go north. But it's an area that it's certainly even Houston per se, it's really fast growing. And it's fast growing because, of course, Houston, it is a massive port. It's a massive international, you know, in and out. It has very diverse also urban and maybe peri-urban spaces. In fact, from Houston, as much as Texas is a big state, you know, a couple of hours outside of Houston and you're really in the middle of farmland and cattle ranch land, right? We have a huge ecosystem of oil and energy, of course, because of the coast. Big NASA, you know, we have a problem, right? Houston, we have a problem. But for us, it was very attractive to move there because we really get hit by some amazing extreme weather disasters, from polar vortexes, right, where we never seen snow and all of a sudden we have, you know, maybe the 5-minute snowstorm, but albeit a snowstorm, to amazing hurricanes and even just disasters. So it's very unique area. And that's why I'm going to tell you a little bit of that interplay between all of these, you know, systems and pressures and stressors and climate, population density. I think we now are probably the third largest city after, you know, we battle with Chicago, but after LA and New York, I think, you know, we're fast growing. So I also have to show you where I work. So I work at the Texas Medical Center. It's smack downtown of Houston. We have at least 250,000 employees that come in and out the Texas Medical Center every day. It's a compendium of 61 medical institutions, all in the area of health and medicine. So this is, it's a footprint that was built specifically for health and medicine. As you can see, we certainly have the largest children's hospital, and also talking a little bit about cancer, we have a whole nexus between MD Anderson and Baylor and Texas Children's. We really tackle a lot of very important health issues. I work at Baylor College of Medicine. That is what I consider my academic hat. And then I also work within the Texas Children's Hospital, which is the alliance with a health system. Right now, all these hospitals are not just hospitals, they're systems. But at the end of the day, I actually am fascinated about a lot of bugs, a lot of parasites, bacteria, fungi, viruses, you name it, that are really what we consider are the highest the diseases of poverty that causes the highest of the morbidity. So maybe they don't kill a lot of people, but they clearly cause a lot of burden. Now the WHO actually categorized these are 17 tropical and now even around 20 emerging infections. They're very prevalent among the poor people. They're very poverty promoting, but don't confuse, right? They don't necessarily only, survive or appear in the poor populations in low middle income countries. In fact, there was a recent paper that my co-partner Peter Hotes wrote that, a lot of these diseases are actually found in poverty pockets amongst the high income countries. That's why also we kind of like Texas at some level, because there's that dichotomy of poor and rich, right? Very ancient diseases. These are not new, very chronic, very stigmatizing. They actually cause a lot of non-visible, sometimes disabilities, a lot of intellectual growth delays, sometimes, of course, disfigurement and blindness. But I think the other thing that is quite interesting is that we forget that if you have these populations with all these parasitic and all these bugs, they really are major cofactors, not only for other infectious diseases, certainly malaria, TB, HIV, but also some of now the chronic diseases like diabetes, and even sometimes more of like the cancer, and certainly the diseases that are affect neurologically. They cause a lot, again, lives lost because of disability. Now we know, we work with a lot of health economists to quantify how much we are losing in economic productivity. And I think one of also the very interesting is that it's not only for humans, right? These diseases are really capturing the whole One Health ecosystem. So I'll talk to you a little bit of a hodgepodge of little things about, all this complexity. Because what we ended up doing when we moved to Baylor, and we've been in Baylor, the group, our entire group has been in Baylor for around 14, 15 years, or close to 15 years. So we created an actual school. We call it a National School of Tropical Medicine. You here in Europe maybe know a lot about the London School of Tropical Medicine and Hygiene, or the Liverpool. So we try to model our school with arguably the hundreds of years that these European schools already have, but primarily also taking a clear look within the Latin American Caribbean sector, even though we work globally, but we were very interested in kind of replicate how we can help, our region. So within this school, we created a model, and I'll talk to you a little bit about certainly our partnership model and how we do it. But we try to integrate a little bit of everything, right? I mean, nowadays, we heard yesterday at the panel how we can be more integrated. So we do a lot of integration of technology work, certainly molecular biology, technology work, with linking it to be very keen of understanding how it can be transferred into understanding how we can serve the population, so therefore population health. I'll tell you why regulatory and policy and finance and access strategies are important for us. And then we do all sorts of things, right? We do vaccine development, which I'll focus primarily most of my talk, but also we do a lot of vector control, molecular epidemiology. We also do clinical, we serve by having a couple of clinics. We do a lot of transmission dynamics. We do mapping, and we, of course, train. We try to train as best as we can the next generation of scientists. So in fact, when we came to Texas, the first thing we did is, what do we find in the region? And to be very honest, we found a lot of stuff, right? I mean, we found a lot of West Nile virus, as you know. In fact, there's a huge cohort of populations that now we've been following with one of our colleagues for more than 15, 20 years within the state of Texas. Of course, we found parasites that people thought maybe we couldn't find, for example, leishmaniasis, Chagas disease. And albeit many tried to ping it to migration and immigration, we actually found local transmission. We have the vectors and we have the parasite, and therefore we find local autochthonous transmission. Murine typhus, we have cysticercosis, toxocariasis, a lot of parasites and a lot of viruses. And in fact, we have now data from our clinics. We have a pediatric clinic and an adult clinic. And as you can see, just by looking at the colors, right, we can find an array of parasitic diseases, fungal diseases, bacterial diseases, viral diseases. Oops. Oh, there you go. Sorry, went away. So we have plenty of work to do, and also plenty of populations that we really would like to help. In fact, one of the latest things that we got is this, again, interplay with climate change, as you know, living in Texas with all the changes. So we just got a prize, in fact, to using some metagenomic approaches to try to create what we call the parasite atlas and virosphere, the Texas virosphere, by looking at predictive models to see if we can identify not only, of course, emerging viruses that appear or disappear. We use a combination of wastewater. We collect, metagenomics from ticks, from redovids, from mosquitoes, from everywhere. And we're trying to, come up with creating this atlas. But today, what I really want to talk to you about is our vaccine center. We call it very easily a long name called the Texas Children's Hospital Center for Vaccine Development at Baylor College of Medicine. If you guys can come up to me with an acronym that would make it easy for me to say this, please. We struggle a lot of how to call it because both institutions want their name on it. So we are, the BCMTCH. And so we've been developing vaccine technologies that we would like them to be very low cost, of course, very safe, very effective, but very accessible. So as you can see in this slide, we use a partnership model. It's called Product Development Partnership Model. It actually was a model that was launched 25 years ago, probably from the Gates Foundation when they started to give funds to try to advance not only vaccines, drugs, diagnostics for malaria, TB, HIV, and certainly NTDs, which is premised on, of course, leveraging resources amongst public, private, for-profit, non-profit, academic, industry, governments, multilateral, partnerships. As you can see, they really, pressured the community to try to advance discoveries into products using this concept of partnering, with also an anchor on open science that we arguably, try our best that we can to share our knowledge, data, reagents. We specifically decided to do a multiple disease focus, but with some limited technology platforms. We use recombinant protein technologies, RNA technologies now for the last maybe a decade or so, and then play a lot with what we call formulation science. But I think one of the very interesting, and a lot of people always ask me, is like, how do you convince your institutions to be creative and have very intentional intellectual property management practices, and how we can incorporate social and advocacy and policy strategies as we move along our projects. So I'm going to spotlight a little bit what we did with the COVID-19 technologies. I don't want to spend a lot of time because I want to give you a much more interesting example of one of our vaccine products. But I think Nuno said that we developed a technology that led to two global vaccines, Corbe vaccine in India and Indovac. We delivered 100 million doses combined. But the way that we did it is with this model, and please, and I want you to kind of try to maybe remember it, because this is our anchor. So we are in that blue box, right? So we do a lot of discovery, like many of you do, trying to find targets. And then through some process of what we call translational pathway, from bench to clinic, we try to apply what we call this first-in-human model, which is very rapidly try to transition a discovery into a product that can rapidly be not only manufactured, but, of course, gets approval to go into a first-in-human clinical trial to establish safety and immunogenicity, maybe take it up to the phase 2, which is proof of concept of some efficacy. But then it's going to be very difficult for an academic center to really push it all the way to, of course, delivery. So we needed to understand who could our partners be. In the neglected disease world, and our date also in the COVID world, we actually were not very lucky of getting big pharma to come and join us because we were trying to develop a low-cost, affordable, non-monetary driven product. So we actually ended up finding out that the best partners for us were the so-called developing country vaccine manufacturers, which actually there is a big network. And if you really don't know this, but the big pharma rely on this network to actually make their own vaccines with their brand, but it's made, let's say, made in India, made in Indonesia or Thailand. And that's the difference between these companies, these manufacturers, to do their own innovation versus just being follow-on producers, right? Somebody hands over a recipe, they have to replicate it. They put the brand of GSK, and then, of course, they're sold around the world versus their own brand, their own innovation. So with COVID, we actually confirmed that the local innovation can happen. And then they have a very difficult task to not only advance the product, move it into the very advanced clinical stages, work with all the policymakers, especially the World Health Organization, ensure they have the highest quality, pass the pre-qualification, then do additional studies, and then at the same time work with the countries, right, to see if there is financing, procurement, and eventually uptake and introduction, right? So that's how actually we did our COVID. And we did it based on 3 things that we learned throughout our 25 years of work in vaccine development. First, we needed to understand that we had to use regulatory science such that everything that we did in our labs could be easily transferred, technology transferred, and that the companies that would receive them would very rapidly be able to incorporate them into their regulatory dossiers and filings. And that's a lot of work. And if you're interested in knowing what that means, it's, documentation, right? Documentation, if you don't write it down, it doesn't exist. And if you don't write it down correctly, it doesn't probably exist either. So a lot of documentation. The second is that, developing the scientific premise of a product is probably the easiest part. The most difficult part is to sell it as a business proposition, is to ensure it fits into a societal perspective. You know now a lot about how people hesitate, maybe are not clear whether it's beneficial to them. So it's very complex, right? So understanding the decision-making process, I think we heard it in the panel yesterday, the social, the political aspects. As much as your discovery is, fantastic, it may not necessarily arrive to where you want it to go, right? And then the third is what I mentioned, right? That in our case, we were really interested in making our products what we call public goods, so really benefit society. And so we really needed to be very intentional with our IP management. And in fact, that's how we ended up having these 2 vaccines. So we, first of all, scientifically, we had a lot of research that we were doing 10 years prior because we were working on SARS and we were working on MERS. So the scientific premise was easy. We had it, right? Then we had to convert it rapidly into a COVID platform, which I have to say it, because of the type of virus, it was not very difficult. But then the difficulty was how to establish these partnerships. We did a lot of what we call evaluative material transfer agreements with option, terms, where if the evaluation worked really well for these companies, then we would do the formal technology transfer. Our anchor of philosophy is always do non-exclusivity. We don't do things that are exclusive because we want to be able to give it to many. We had no patents, so the real IP was really our know-how, our recipes, our reagents. And then we worked with, of course, two companies that had the same philosophy that we did, right? That they didn't want to jack up the prices of these vaccines, that they really want to make them to really distribute them. And so that was, of course, in our agreements that, they had to have some global health pathway access for us. So it worked really well. I have to say, if you want to hear about how our experience is to work with these large industrial manufacturers, we can talk, certainly during coffee break. And if you're even interested in what it means to develop a halal certified vaccine, also come and talk to me, because that is, again, documentation. So it's a fascinating story. If you scan the QR code, I think it still works. You can read an expert review that we wrote in vaccines that tells the whole story of how we got to our COVID vaccines. But our portfolio is larger than that. And in fact, here's just a snapshot. We have a lot of interesting parasitic infections, hookworm infection, schistosomiasis, Lyme disease, Chagas disease, which we've been working for the last 25 years. And albeit we managed to do the COVID work with our 10-year prior record, but when we transfer everything to India and Indonesia, it took us less than a year to get to delivery. That's impressive, right? We are still trying to navigate how to do that for these neglected diseases. So I'm going to actually tell you some trends that are happening globally and how now we're reorganizing ourselves to try to fit into these frameworks. Because unfortunately, as much as we want to advance our scientific discoveries, we have to rely on what would a manufacturer really want and need, what are the criteria in the policymakers, WHO and others, and what's the economic and the demand that we need to respond to. So it's not as easy as just continue working with our nice recombinant technologies or even RNA or protein or viral, which of course are still quite in fashion. Using adjuvants is important. You've heard now that it's not as simple as just trying to do intramuscular injections. Now people are really looking at alternative mucosal patch technology. I'll tell you a little bit about what controlled human infection models are and how we actually incorporate it into our scientific strategy. Yesterday, we heard a lot about AI. That's really helping us a lot in target, screening and identification. The fact that we need to also shift a little bit our terminology. I mean, some people don't like vaccines anymore. We have to use either directed therapies, immunomodulators. So there you go, right? And we have to, again, I'll tell you a little bit about the DCVMNs again, and then this concept, right, of what are these multi-decision criteria? Because if we do all the work in our lab and we don't pay attention of what these policymakers are trying to advance, then you get stuck with something that goes nowhere, right? And I'll use, in fact, the combination vaccine trend. This is brand new. It's coming up, and it's like now everybody is aspiring that now there's really a lot of concern by parents, by, programs in countries, to more vaccine injections, more, right? How many schedules, how many doses, right? And so they're really trying to look at how can you mitigate overloading this schedule, right? And how do you do it scientifically sound and safe, right? The procurement and the implementation is complicated, right? We saw it even when you go from even pediatrics to adults, right? So combination seems to be some solutions. So in fact, WHO is now really moving into creating a framework, and God forbid you're not on the list of how you fit in that framework. We're never on the list, I have to tell you. But they are trying to figure out how to do it. So I'll tell you the case study of hookworm and malaria combo. Because when we started developing our hookworm vaccine, we always thought it could be a standalone hookworm vaccine, because there's plenty of people that would need it. It definitely causes a lot of anemia. But then we started understanding that because it's a blood-feeding worm, it causes, of course, anemia. It's co-endemic with malaria. It's actually additive anemia to malaria, that maybe we should never forget that it could be at least either co-administered, but arguably potentially even co-formulated with malaria. So actually, this actually struck a little, interesting when we were talking to all the WHO people, right? Because it's not only interesting, but it, maybe scientifically could also be very interesting. So that's what I'm going to tell you, what we're doing. So first, I need to tell you what is hookworm. And I'm sorry I'm grossing you out a little bit with that video before we go to the break. But again, so it's a blood-feeding worm. It's very little. It's a 1 centimeter little worm that lodges into your intestine. But it feeds so much on blood that, in fact, from only 25 little worms, you can deplete the entire iron, daily iron intake of a child. And usually you don't have 25 worms. Usually you have burdens of maybe even hundreds, right? Depending on the transmission area. So with Alex Lucas, who used to work with us, now he's at James Cook in Australia, we actually decipher, and this is where all the molecular biology comes in, we decipher the entire feeding pathway of what happens when the blood enters into the gut of the worm, right? So it's the feeding pathway inside of this little worm. And we found that there are very, very orchestrated series of events that uses not only a combination of aspartic proteases, serine proteases, metalloproteases, that chops up the hemoglobin. And eventually, all this heme that is being produced also has to get detoxified, right? Like we go to the bathroom, right? We eat, we digest, and then we go to the bathroom. The worm does exactly the same thing. So we tried pretty much for the last 25 years, every little candidate to see if we could find some that seem to be doing what we want, which is to block the blood feeding. By blocking blood feeding, especially in the female worm, you also can reduce fecundity. So there's less eggs in the feces, and therefore maybe we could not only reduce the burden of disease, but also interrupt transmission, okay? We actually tried all of them. We found 2, but today I'm going to focus on 1, which is the detoxifying agent. Is a hemoglobin binding and detoxifying glutathione S transferase. We discovered it in 2005. We actually make it using a Pichia pastoris, a yeast expression system. It's a small protein, 24 kilodaltons. We then formulate this protein with a aluminum hydroxide, and then we play around with immunostimulants, toll-like receptor 4, toll-like receptor 9, toll-like receptor 7, all these toll-like receptors to see if we can bolster not only the immunogenicity, but also the potential protective ability of this molecule. We did a lot of studies in preclinical animal models. They all looked really promising. But then we had to move into the clinic, right? So we did, in fact, a series of first-in-human studies. We've done them in the United States, in normal, healthy, non-ever infected people. We then moved to Brazil in previously infected populations. We then jumped over to Africa. We also did some studies in Gabon. So we de-escalated in children. So we know that it's not only safe, but also certainly immunogenic at all ranges. But then we had to very rapidly figure out how to do a proof of concept that it works. And doing field large phase 2 trials not only cost hundreds of millions of dollars, but it's complicated. So we ended up developing a control human infection model, which means that in a control setting, in actually a clinic in GW in Washington, D.C., we would bring some cohort of individuals where we would vaccinate them, but then we would challenge them in the lab with a control number of infective larvae, of course, all under regulatory dossiers. And then we can determine, right, if those that were vaccinated versus those that were not vaccinated, if you actually can see an infection, and we look at infection by quantifying the number of eggs in the feces. And after a certain amount of time, fortunately, we can treat them and we can give them, albendazole, and they can go home and live happily ever after, right? We actually managed to do this, and we tried various formulations. We tried, again, our GST molecule with alum plus these toll-like receptor agonists. And there was one, which was the one using CpG, which is a toll-like receptor 9, that gave us 100% protection. So as you can see in the little chart here, the placebo, you can see eggs in the feces. Some of the formulations certainly looked promising, but not fully sterilizing, but the purple line at the bottom is the formulation of alum, CpG, and the GST protein. So we got really excited about this. We didn't think we were going to see any efficacy because it's only 25 little larvae that we actually infected with. We can use 25 or 50. We've gone up all the way to 75. And we could not detect some patients, we just could not detect any larvae. So this was really impressive for us, right? So what do you do next? That's the big question. That's where the complicated aspects come. So first we needed to then create a business case. So we actually tried to get partners to help us decide, is there a market opportunity? Do we have a licensure path? Do we have a financial model? Who will be our partners? Again, developing country producers. What's the structure and the funding path for them? What other preclinical work do we want to do if we want to incorporate it with malaria, for example? And then we needed to work with all these policymakers, right? And I think, what we ended up realizing that there actually could be a very enticing potential market if we manage to co-formulate it with the malaria vaccine candidate, because we found that where the top malaria demand is currently right now, and we have 2 malaria vaccines, actually coincide with the top 10 countries that have the highest co-endemicity with hookworm. So we definitely have a market. And in fact, we projected that in this market of these countries, which as you can see there, could be from Nigeria, DRC, Uganda, we actually can position a sale of around 70 million doses. And that 70 million doses can actually recover around $300 million in market. So maybe if you were a developing country manufacturer, maybe you say, okay, maybe I'll invest into this. So we're really interested in seeing what happens when we present some of these ideas. But I just want to wrap up with, the fact that the way we did it and we do our projects is that we really rely on academia connected with a health system so that we can really move the innovation, right? The technology innovation. You have to partner early with whoever you want to hand over your discoveries. In our case, the developing country manufacturing network is a key stakeholder for us. We also realize that it's not only science, that you need to understand the social and business strategies behind it, and you have to align it. There's no way out, right? I mean, we have no option. And for NTDs, meaning all the vaccines that we're working on, it's very complicated, right? Because it's challenging, what traditional pathways are just not going to work. So we really need to look for, again, what's the trends? And I think combination vaccines may be one of the potential solutions. Funding is always a challenge. Policy prioritization is always a challenge. But we're open for new partnerships. So, anything that is, of potential interest to us, we're definitely going to advance it. this has been done by an amazing number of people. This is our little group in our vaccine center. If you look at the sponsors, I really want to bring your attention to one very special sponsor, which is, how many of you drink vodka? And how many of you know about Tito's vodka? I don't know here in Europe, but in the US, it's a big deal. Tito's vodka actually is made in Austin, Texas. And Tito's actually was one of the funders that really is helping us advance these innovations. So when you think about getting funding, don't always think on the most logical funders of research. Look for the very alternative funders of research. But I thank you very much for your attention.
Very few questions. 2 questions at the most. Questions, please.
How, I don't know if you can talk loudly, how does DCV, what is it called?
DCVMN?
DCVMN fund itself? In other words, it's manufacturing and then not charging a lot. So how is that?
So it's volume market for sure, right? Volume. They make. For example, the scales that they can make, it's incredible, right? In fact, that's the reason why the big multinationals, they don't manufacture. They don't have their own manufacturing anymore, right? They transfer to them, and they're their CDMOs, right? They're contract manufacturers, and then for their own countries, certainly India is enormous amount of population, right? Indonesia, it's arguably also the Muslim majority country region. So it's really a volume market, really a volume market.
Can I ask?
Sure.
Okay. Concerning the COVID vaccines, you didn't talk about clinical trials, and when were they produced compared to the RNA vaccines? Can you?
So the clinical trial plans were all driven by the manufacturer, right? So we handed over that technology, they matured the technology, they produced the doses, of course, and then in India, they did the studies in India. In Indonesia, they did the studies in Indonesia. So each of the manufacturers had to take on their own clinical development plan. It took them around 12 to 18 months to get the first emergency authorization in their countries. Unfortunately, you know how long it took Biological E to get WHO pre-qualification so that it could get the vaccine exported? It took them 5 years.
But these doses that you have mentioned, when were they used?
They were used in their own countries.
Yeah, but when?
They were used right when they needed the vaccine. So for example, the BioE, I think they started to get deployed in mid-2021, like May 2021.
But the 5 years that you are mentioning, that was done.
From 2020 to 20, yeah, they just got the pre-qualification stamp. And of course now, nobody, they're not going to export it. They're just using it for their own local. It took WHO too long to give them the authorization for export.
So if I may, since you sort of handed an option, how did you get the halal?
The halal was really based on documenting that nothing in the process and reagents had any animal-derived materials, and especially porcine-derived materials. So we actually got monitored and audited by their Ministry of Halal Readiness, like, religious ministry of, that they came and they looked at all our records and they showed that we had no materials, everything was synthetic, everything was, not of vegetable origin.
In the cases where you have something you want to then partner with, take it to the next step, do you have specific criteria which have to be met to make that decision, or do you just have a board which gets together and decides how to?
We look is, of course, technology, right? So even amongst these big network of manufacturers, not all of them use recombinant protein technologies, for example. Many use, inactivated or viral or whatnot, right? So what we did is we first do a landscaping of who makes hepatitis B vaccine, because hepatitis B is made as a particle, not as a monomeric protein, but it uses pichia or yeast-derived processes. There we kind of narrowed it down to, I don't know, the top 10, let's say, right? And then we looked at who has a philosophy of being a little bit more altruistic and really keeping in line with global health. You know, I've known all of them for years, right? So we started doing our little rounds. And in fact, BioE, which, by the way, if you're interested, it's a woman-led company. So it's really very impressive what she has done. It's probably the third largest in India after Serum Institute and maybe Bharat. And they were the ones who really genuinely looked at the technology. They wanted to have their own homegrown rather than working again with AstraZeneca or working with J&J, or right? And so that was clear. And then, of course, when our licensing office was negotiating, they were really amenable of not shelving our technology, because some people may get your IP and then just shelf it and not do anything with it. But really, we had terms that they would really push it. And, you know, even if they didn't get to the end, but it clearly that they had the best effort to keep it accessible and affordable.